Hello, everyone. Let's finish up with the reactions in gluconeogenesis in this video. So we saw how we bypass pyruvate kinase and how we can, as we've discussed in the past, how all these other reactions are reversible. So we are now stuck at the fructose 1,6-bisphosphate step, which is in the way it goes in glycolysis from left to right. We cannot go from right to left. It's irreversible. So we need to figure this out and how that will happen. So we're going to remove, actually, let me go back. So both in this reaction and the next one, where if we need to bypass hexokinase, where we have to remove the phosphate on the 6th position, we're going to have phosphatases to do the job. So phosphatases are thermodynamically very favorable. You hydrolyze the phosphate that you carry. They're specific for a specific substrate and the phosphate and the position of that substrate, right? So there are very unique phosphatases, and the hydrolysis of phosphorylation is generally thermodynamically favorable. All right, so we've seen this already. FBP is a reaction that very special for this particular step of reversing, in a sense, the PFK reaction. And what's interesting, so it removes the phosphate on one position of fructose 1,6-bisphosphate to generate fructose 6-phosphate. What's special about FBP, or not special but intriguing, is that it is regulated allosterically in the opposite way that the same allosteric effectors regulate PFK. So, high AMP, which means low energy status, okay, will activate PFK. That makes a lot of sense because you have to take glucose down through glycolysis and burn it and make some ATP, let's say. Another allosteric effector is fructose 2,6-bisphosphate. But if you look at the effect of AMP, for example, it's completely opposite on FBP. So you have high amounts of AMP, you will inhibit FBP. Does that make sense? Well, who wants to do gluconeogenesis when you don't have enough energy, right? So first things first, you have to get your energy to use in biosynthesis. So if you don't have enough energy, you cannot do biosynthesis, essentially. You cannot do anabolism in this case. So that makes sense. So then usually in most cells, this is where it stops. So with a generation of from fructose 6-phosphate, of course, with the glucose isomerase, you can go back to glucose 6-phosphate. And this is where it stops because if you get to use the glucose inside your own cell, you don't want to dephosphorylate it because it will go out. However, if your job is to actually make glucose and send it out to other places, then you have to reverse also the hexokinase step, and you have another phosphatase that will remove the phosphate on 6th position of glucose, and you got free glucose. So that's easy. It doesn't usually happen in most cases, but in humans, it happens in the liver. Okay, that's the job of the liver, and that's something we're going to see in the next slide, in the next video as well, in the Cori cycle. But in humans, some other tissues, like, they're very greedy. Okay, brain and muscle never give their glucose to anybody else. So they don't make free glucose. They will do gluconeogenesis to make glucose 6-phosphate, and that's where it stops, just like microbes. So the overall reaction of gluconeogenesis is shown here, and I draw your attention to energetic needs that you've spent. Again, this is an anabolic pathway. You make stuff from smaller things, and what you need is energy. So in ATP and GTP in the PFK reaction where we reversed it, okay, to go from oxaloacetate to phosphoenol pyruvate. And of course, you need reducing power in the form of NADH too. So these are the usual goodies you need for every biosynthesis, and gluconeogenesis is not an exception. If you want to see where you used all these steps, or all these ATPs and NADHs, here they are. Okay, so pyruvate carboxylase and phosphoglycerate kinase, and then you have PFK with the GDPs. Everything gets doubled because if you want to go to one glucose, you have to use 2 pyruvates, obviously. And of course, in the GAP-TH reaction, this is where the redox part of the lower part of glycolysis is. So that's where you're going to need the input. So cells have to pay for it. All right, so we're going to stop at this point, and then the last video of gluconeogenesis, we're going to discuss the Cori cycle.